Hello and welcome to another Royal Society video podcast. Today I'm joined by Professor Roy Quinlan at the University of Durham to discuss his latest research on the effects of low-dose radiation on the eye lens. So Roy, what do we know so far about the effects of cataracts and X-ray exposure?
So cataract is an iconic problem associated with exposure to X-rays. And it goes way back to when X-rays were discovered. Within a few years it was known that actually the eye was very sensitive, and by 1931, with the work from Schneider, they knew that the lens, rather than the retina or other tissues within the eye, was very sensitive to radiation damage. The consequence is that you get a cataract, and a cataract is a clouding. It causes light scatter so that people will no longer be able to see, and you can, in severe cases you can see that the lens is white, so light doesn't get through. You need to appreciate some of the biology of the lens, and I've got a little prop here which I'll show you just to help us understand. If we assume that this is our lens, the lens epithelium just covers the anterior pole of the lens. So light comes through the cornea and then through this central part of the lens, passes out the back on its way to the retina to form the image. And where this bit of tape is where we would say is the proliferative zone, which is where the cells are actually going to be dividing. But this body, this volume of the lens is made up of fibre cells, lens fibre cells, which are differentiated from these cells. Now, in order to maintain this perfectly round shape, it means that all of the cells at this perimeter have got to divide or produce fibre cells at the same time. So when you begin to disturb the proliferation around in this region and you increase the cell density around here, somehow the lens is going to try and adjust to this, but it does it imperfectly. So then 10 months later, these mice that we followed in this low dose range, as I say, peaking around about the 350 milligray, they had lumpy lenses. The lenses were misshapen. So we can follow the effects at 24 hours where the density increased right through to what happened at 10 months. But to be fair, DNA damage is usually repaired very quickly in cells. And you can follow that by looking at markers, for instance, gamma H2AX. And we measured the response to those for those cells, both in the central region and in the peripheral regions of the lens. And the interesting thing there was that the response is linear. So when you provide low, 20, 50, 100, 200, you can draw a straight line there between the foci of gamma H2AX that are produced. Okay? But interestingly, the cells at the periphery, they didn't repair as well as those cells which are in the center. And we made a comparison to circulating blood lymphocytes, because that's the gold standard in radioprotection. If any person is exposed, then you take a blood sample, and measuring the number of foci that you find in the circulating blood lymphocytes, you can actually, and knowing the time of when they were exposed and between the time that you're actually doing the measurements, you can actually tell what sort of dose that person has been exposed to. So we made that comparison, and basically those central cells they're professional, they know what to do. This is DNA damage, and within 3 hours it's goodbye Jose, they're finished, they've done it. Just the same as the circulating blood lymphocytes. But those cells at the periphery, they had not repaired their DNA, and even after 3 hours there was still considerable amount of DNA damage that's maintained there.
And so you mentioned obviously the effects on the mouse lens. How does this, and how can we relate this to kind of human use and its importance?
Yes, of course. Yes. Well, there's the legacy of the A-bomb survivors, and of course for that and Fukushima, you don't have to look very far back in history to see the problem of radiation exposure, and at those high levels, at those doses that were experienced by cleanup workers, for instance. Chernobyl is another instance. You need to know and you need to know what to look for and where to look within those patients so you can monitor them and help pick up problems that they may experience. But surprisingly, it's now we're realizing that the thresholds that we've been talking about for the lens are very much less. So suddenly it brings into focus those people that use X-ray imaging, medical imaging. And there's reports in the literature, epidemiological studies with cardiologists, for instance, show that if they don't take the necessary precautions, they have cataract. And it goes beyond that because, and again, another very specialist group of people where this is very important are astronauts. It's been long known, NASA have invested quite a lot of money in looking at the effects of radiation damage on the lens and on the eye. And as sure as eggs are eggs, if we go to Mars, they're going to have to find a solution to this, or else they're going to have to find a Specsavers when they get there because those astronauts are going to have problems. So you can see, for that group, it's a problem, and for radio imaging, it's an issue as well because 20 millisieverts it's not a big dose, and I think as scientists we have a responsibility to society to actually work out what's happening and why these are happening, so that we can respond appropriately with therapeutics or ways of preventing this sort of thing. And as an example, the lens and proliferation, I've got to return to this because this again it's fascinating. The circadian rhythm that we all experience plays a role here.
Yeah.
So early in the morning, so we know about that, and we took that into account when we did our experiments. So all of the animals were exposed in the morning, because in the morning is when this burst of proliferation occurs.
Oh, that's interesting.
So you see, the daily cycle actually impacts what happens, and that's been known since 1952 again, or the 50s from von Salman's work, where he looked at mitotic indices in rabbit lenses, and he said, AM much higher, PM lower, and so that PM is a time when there would be less sensitivity. So it leads on to the questions. Well, what if we actually expose animals later in the day? Do we see a different response? And then we know that actually with age, the proliferation rates drop. So should we send old people to Mars? So it gets along those sort. But with the experiments, you does age have an impact upon this as well? Because is it the ability of these lenses, or the feature of these lens epithelial cells to divide and be proliferation ready? Is it that which actually predisposes them to being more sensitive? Or is it that their DNA repair pathways are actually severely compromised?
And what would you say are the next steps in your research?
It's got to come down to mechanism. We need to understand what it is about the DNA repair pathways present in the lens epithelium, and how that predisposes the lens and that particular part of the epithelium to the problems that then arise, increased cell proliferation, and then the issues associated with the misshapen lens.
Thank you very much for your time, Roy.
That's a pleasure.
Thanks.
Thank you for coming to Durham. Views expressed in this interview are my views. I'd like to point out that they in no way represent the views of the Department of Health or the National Institute of Health Research or the NHS, even though the Department of Health gave money in order to support the research reported in the paper and represented here in this interview.